In this module, we turn to the study of inheritance. And I know we're going a little bit out of order. We're moving into chapter 9 after we've done chapter 10. But I like the idea of introducing the study of inheritance after we've talked about the function of genes. And remember that the function of genes is to encode proteins. And it's the action of proteins that actually determines the traits that we think of as inherited traits. And there are plenty of traits that we think of or that we don't think of as an inherited trait that are also controlled by the proteins that are encoded by genes. Gregor Mendel, an Austrian monk, published his work on the study of inheritance in 1865, and it was ignored for a long time, and no one believed Mendel until the early 1900s, like 1901, when people really started to figure out the cytology or using the microscope to figure out the mechanism behind meiosis. So we're going to see that Mendel proposed that everybody had two factors for a particular trait, and those would be the two versions of a gene. He proposed that in producing gametes, those two factors separated from each other. Those would be the two alleles or alternative forms of the gene. And then each kid was given one allele or one version of the factor so that when the offspring were produced, they again had two versions, one from each parent. So this was proposed in 1865, well before meiosis had been worked out. And before that, people didn't really understand what it was that was inherited. They just called the study of inheritance or the theory behind inheritance the blending theory, where there was some factor that was contributed by both parents and those two factors blended together to create the trait in the offspring. Mendel used a very systematic approach to study traits that were known to be inherited and very carefully determined how these traits were inherited from one generation to the next. So this first lecture is just an introduction into his genetic system. His genetic system was based on the pea plant. The pea plant is a diploid eukaryote, of course, because it's a plant. And so there are two copies of every chromosome in the nuclei of cells of the pea plant. This plant is self-fertile, which means that in the flower, which is the sexually reproducing organ, there are both male sex organs and also female. At the base of this structure right here, there are haploid eggs produced, and at the tip of this haploid structure, stamen, there are haploid pollen produced. This plant can self-fertilize itself, haploid gamete fertilizing another haploid gamete, or the plant can be cross-fertilized with another plant of the same species. And so this was important for Mendel's work, to be able to do both self-fertilization of a plant and also cross-fertilization between plants. Cross-fertilization involved taking two plants and on one removing the stamen so that the plant was only female, then taking pollen from another plant, the male gametes, and fertilizing the female structure of the original plant. In that way, like in this example, the female acted as the purple plant acted as the female, and the white plant is acting as the male. The male gametes are transferred to the female gametes. Now, in pea plants, the developing peas are the offspring. These are the now diploid offspring that have been produced from a mating between two plants, or it could also be a mating within one self-fertilization. Those peas are then planted and they grow up into individual plants that are also the offspring of the original mating. Recall Mendel's first matings where we have one male and one female, two different plants. We call those the parental generation or the P generation. We then call the offspring of that mating the F1 or the first filial generation. He took plants from the F1 and self-fertilized them. Sometimes he cross-fertilized between plants. But the basis for his work was either cross-pollination or self-fertilization, using gametes from two parents or one parent in a self-fertilization and producing offspring which could then grow up and the traits analyzed. The 7 traits that he looked at were flower color, flower position, seed color, seed shape, pod pod color, and stem length. These were 7 traits that were known to be very well inherited, stably inherited from one generation to the next. And on the left-hand side of the 2 traits, this was the version of each trait that was known to be most prevalent. This was the version that was known to be less prevalent in the population of plants. He called that most prevalent trait the dominant trait and the least prevalent the recessive trait, or the recessive version of the trait. For example, purple flower color is dominant to white flower color. The word trait is kind of loosely used. Sometimes it's referring to just flower color in general. Sometimes it's referring to the distinct versions of the trait. And that's what I prefer to say, that there's a recessive version or a white version and a dominant or purple version of a trait. The word character is hardly ever used, but this is the overall idea, that there were 7 basic traits, and for each one there are 2 alternative versions. Now, notice that there's no in-between version. There's no light purple version. There's no light green version. There is a very specific dominant and very specific recessive trait. We're going to look at how Mendel used his matings to analyze the inheritance of these traits. We won't talk about the inheritance of all the traits because the inheritance was actually very similar for all 7, but we will look at a couple of them in the next few lectures.